Well, it really is an enormous honor to be here. Thank you, Bruce, for the introduction and the opportunity to speak to all of you today. And I'll just reiterate what Bruce said, which is there are some of the most spectacular scientists in the world participating in this meeting, and I'm really deeply honored to have the opportunity to tell you about, as Bruce said, the topic du jour. Now, let's see. That topic is revealed by this first slide. No one becomes obese unless they eat more calories than they burn. But that conclusion would ignore the deeper question as to why they eat too much. And that's the single question that has captivated my research for the last some number of years. Views on this may have changed with the development of the new therapies I'll tell you about. But historically, there have been three general explanations for why some people might eat more than others. One is that the obese lack a level of willpower normally exhibited by lean people. I find that point of view is more often favored by lean people. A second possibility is that we live in a modern environment where people don't exercise and have free access to calorically dense calories. And then the third is that there are biological and genetic factors. And I'm going to tell you about biology and genes today, but also tell you that I used to take a vote and ask people before and after I spoke which of these three they favored, and I stopped doing that when fewer people voted for biology when I was done. So I'm not going to do that, but rather what I'm just going to point out to you is that if I were talking about any other disorder, people would gravitate to the idea that there are biological factors on which volitional factors and environmental factors operate. Think of heart disease, think of cancer, and so on. And for reasons that are more sociologic than biologic, people tend to have been gravitated to one explanation to the exclusion of the others. So I'm going to tell you something about what causes the problem in the lecture today, which is named in honor of Dorcas Cummings. But I put this up in also part to acknowledge and thank the donors and to tell you from an outsider what a fantastic institution this is. In addition to the inestimable impact on biomedical science, these meetings are legendary in the field of science and have been the center of scientific discourse and communication for almost a century. I'm also pleased because I grew up on Long Island. The X is where we are now. I grew up on the South Shore, literally on the other side of the tracks. But of course, I knew Cold Spring Harbor growing up and would visit here on occasion when I was younger. I'll also point out to you that I'm a Ranger fan. I suspect many of you are Islander fans. But even if you are, I'm hoping that you'll give me your best wishes and the Rangers your best wishes for tonight. Okay, so this is the topic of the presentation today, the causes and treatment of obesity, the end of the beginning. I pointed out in a previous talk that the original version of this said the beginning of the end, which I thought was unnecessarily apocalyptic. And I'm going to tell you what causes obesity. I'm going to tell you a little bit about how we currently treat it and then where we go from here. And the reason I'm referring to this as the end of the beginning is that right now the treatments for obesity don't treat the root cause of the disorder. And in addition, there are a limited number of modalities. And I think as a field matures, once the door is unlocked to new therapies, tailored therapies are developed. Some patients respond better to one therapy than another. And ultimately, the best therapies target the pathogenic mechanism, which is not the case currently. And the best example of this was that I can think of is a talk that was given in 2005 by Charles Sawyers at a previous Cummings lecture, talking about chronic leukemias. And back then, until Charles's incredible work, chronic leukemia was treated with conventional chemotherapy. And it was somewhat effective, but not curative. But in more recent years, drugs have been developed that directly attack the pathogenic mechanism and have converted this cancer into a manageable, potentially curable disorder. So today what I'm going to put forth is the notion that obesity is caused largely by biological and genetic factors, or at least there's a biological underpinning on which these other inputs act. So the strongest evidence for that come from studies of identical versus non-identical twins. These are the world record holders at 1,400 pounds per zygote. That's an inside joke for the scientists. The zygote is a fertilized egg, so each of these twins weighed 700 pounds. And it illustrates the point that identical twins are more alike for their body habitus than are non-identical twins. Here's a systematic example of that. The top are monozygous, the bottom are dizygous twins, and it's immediately evident that the monozygous twins are more alike than are the dizygous twins. And from these systematic comparisons, you can get an estimate of what the heritability of a given trait is. And if you do that, what you find is that obesity is as or more heritable than any trait that's been described, with the exception of height. Higher genetic loading than schizophrenia, diabetes, hypertension, alcoholism, heart disease, all disorders that we commonly accept have a genetic basis. So with this as the starting point, my laboratory was interested in what these factors might be, and our attention was drawn to this mouse, the Obi mouse, identified by George Snell, who won a Nobel Prize in 1951 for another topic. Snell and colleagues noticed that a mouse that was the product of brother-sister matings was slightly chubby at weaning, but over time became more and more obese, weighing 3 times as much and having 5 times as much fat. One of Snell's colleagues told me that at first they thought this mouse might be pregnant, except for the fact that it kept getting fatter, it never delivered, and it was male. And so based on this, they made the leap that this is actually a genetic form of obesity, and the only difference between this animal and this animal living in the same cage with the same free access to food is a defect in a single gene. Now, in 1994, our laboratory identified the defective gene in these animals as encoding a hormone named leptin. And I want to just make sure we're all on the same page here in terms of what a hormone is, because I am going to tell you about more hormones than you ever wanted to hear about in your life today. So hormones are signaling molecules. They're made by one cell in the body. They communicate information about physiology to other cells in the body by circulating in the blood. And they act on target cells through specialized molecules known as receptors. Think about this as sort of like an electronic system where you have a transmitter and a receiver. The hormone is the transmitter sending information. The receptor is receiving that information and then acting on it. So leptin is a hormone made by fat tissue. It's made by fat tissue in proportion to its mass. More fat, more leptin, less fat, less leptin. It acts on a key part of the brain known as the hypothalamus, which regulates nearly all, or perhaps all basic drives to regulate food intake as what is referred to as a negative feedback loop, which I'll explain more about in a moment. But what happens in the Obi mouse is that there's a mutation in the leptin gene. The hormone just isn't made. So no signal is ever generated telling the brain that there are adequate fat stores, which every organism needs to survive a famine and to provide a source of calories when food is not available. So in the absence of this signal, the brain thinks the body is starving. The animal eats more, continues to eat more, make more and more fat tissue, but the system is never shut off because of the defect in this key signaling molecule. So this video explains how this system regulates the amount of fat each mouse or human carries. So at your stable weight, you have a certain amount of fat and a certain amount of leptin, and the amount you eat equals almost exactly the amount of calories you burn. Now let's imagine for a moment that you go on a diet or lose weight or are subject to a famine. Fat mass is decreased, leptin level goes down, and that's a potent stimulus to eat more. And now you'll eat more until such time as the fat mass and leptin level returns to the starting point. Now imagine you spend a week eating at 3-star restaurants in France or McDonald's, whatever your preference is. Fat mass and leptin level goes up. This will suppress appetite, again returning fat mass and weight to the starting point. So what this system does is it keeps your fat mass in a relatively narrow range, which for reasons I won't talk about in the interest of time, is under intense evolutionary selection. So what this does, the system does, is it provides a biological force that resists weight change in either direction. This explains in part why it's difficult to maintain weight loss after a diet, but very few people know that it's actually even more difficult to sustain weight gain over the long term. And it's this system that maintains homeostatic control, as we've referred to it, of fat mass. We keep this biological parameter within a relatively narrow range. Now, as I said a moment ago, this system sets up a biological force, but that doesn't mean that other factors don't intervene. They do. I'll tell you about them. But it means that if you want to that if you're going to change your weight, you're going to have to overcome this biological force by one means or another. Now, to illustrate the relevance of this system to human, I'm going to tell you about a case report that was put forth by my friend and colleague Stephen O'Reilly in Cambridge, England. This child was of normal weight at birth, but began to develop morbid obesity in infancy. He overate voraciously as an infant. He was already pre-diabetic at the age of 4 when his weight was 90 pounds, with 50% body fat. This child had a similarly affected 9-year-old cousin who was 8-year-old and weighed 210 pounds, which is what I weigh. All right, 220, but. When you have an extreme phenotype in an inbred family, which these children came from, they were first cousins, the parents were first cousins, it often indicates that there's a single gene defect. And O'Reilly and colleagues looked, and sure enough, leptin was missing from these children, much the same as they were from the mutant mouse I showed you. So they began to treat the child with leptin, and here's what happened. The child got older. That has nothing to do with leptin. That's just the way things are. Despite getting taller, he got thinner. His body fat went down. His pre-diabetes went down. Insulin level went down. Before the first leptin injection, they gave a child a test meal of 2,000 calories. The child ate all 2,000 calories and asked for more. After a few leptin injections, now the child ate an age-appropriate number of calories at that same test meal. This is now a photo of the child at the age of 8. So what was driving obesity in this child was not a toxic environment. It was not a lack of willpower. It was a lack of the hormone leptin. Now, I should tell you that mutations in leptin are extremely rare. Hormones are very poor targets for mutations. There's a fairly well precedented that mutations that ablate a hormone altogether are extremely unusual, and that's because of what a critical central role they play. But as I'll tell you in a moment, mutations in the pathway that by which leptin acts are not rare. Okay, these children are obese because they lack leptin. What about the rest of the population? Do they lack leptin? And the answer is clearly no. In this slide, what we're doing is measuring the amount of leptin in the blood relative to the amount of fat an individual carries. And what you'll note here is that as fat mass increases and patients become more obese, leptin level goes up. So in general, obese patients have higher leptin levels than lean patients. Well, you might then ask, if you already have high levels of a hormone, what good does it do to give extra levels of it? And in most other hormone-based disorders, the answer is not much. And that's what you see here with leptin treatment. So in the initial trials with leptin, at the highest doses, there did appear to be a small effect in some of the patients, but there wasn't in others. And moreover, the doses that were used in this case were so high as to make it a non-viable commercial entity. So here we have high hormone levels and a reduced response to the exogenous hormone. These are the characteristics of what we call a hormone resistance syndrome. For some reason, there's a block in leptin action. So no signal gets through, or a subnormal signal gets through to the hypothalamus. This then leads to an increase in fat mass, food intake, increase in fat mass, as if by dialing down the response to leptin because of this block, weight goes up and leptin goes up. You're resetting it. Now, that explains why giving extra leptin is less effective. There's a block to leptin action that we believe underpins at least part of the causes of obesity. Now, in some cases, we know the causes of this leptin resistance. And that's because O'Reilly and others have shown that mutations in a series of genes on which leptin acts also cause obesity. That includes not just leptin, but its receptor, which we call leptin receptor B, and downstream mediators, molecules called alpha MSH, and its receptor, the MC4 receptor. And we know that about 15% of morbid human obesity is a result in a single gene defect in one or another of these genes. 15% single gene, an incidence of 15% of single gene disorders causing a complex problem like obesity is as high or higher than that for any other complex trait. So leptin resistance can be the result of a known defect in this pathway, but in most patients we don't know the basis yet, although I'll just digress for the cognoscenti and say that in a recent work we've also found that diet-induced obesity, as it's known in mice, if you feed a palatable diet to mice, they become fat and leptin resistant. And Bowen Tan and Kristen Hedbacker, who was at this meeting, recently did some work to show that this dietary change in mice that causes obesity also affects this pathway, suggesting that there are also acquired defects in this signaling pathway that underpin leptin resistance. And because there's leptin resistance, leptin as a treatment for most patients is largely ineffective. The new treatments, therefore, do not at least directly target the leptin system, although the full range of their actions isn't known. Rather, they target a different brain circuit that regulates appetite. This was a figure published by Scott Sternson, who was at this meeting. And what Scott pointed out is something that's quite well known, is that this homeostatic system I told you about is not the only system that regulates appetite. There are three highly intercalated systems that control how much you eat over the short and the long term. Leptin is part of a long-term system, a homeostatic system, the purpose of which is to maintain your fat within a relatively narrow range. There's also satiety and reward systems. Satiety is the feeling you get when you complete a meal and you're full and don't want to eat anymore. Reward and hunger are driven by separate pathways that drive you to eat, for example, in between meals and keep eating until you're satiated. So the key point here is that during the course of a day, you have what's called a meal pattern. These are signals that determine how hungry you get between meals, how full you get during a meal, and that these short-term systems interact with the homeostatic system to make sure you get your nutrition right, which is absolutely indispensable for all living organisms. The new therapies that have been developed primarily target the satiety system, and some of the players are listed up here, about which I'll tell you more in a minute, and they're based on the fact that another set of hormones made by your GI tract can regulate satiety. So what I've described for you is sort of a short-term system that controls your meal pattern during the day, and a long-term system that modulates these signaling pathways to keep your fat mass at a stable level over the long term. So this simply restates that there's a long-term system controlled by leptin, and then a whole host of gastrointestinal signals that are secreted in response to a meal and that play some role to feed back down-regulate appetite and terminate a meal, either by acting directly on the brain or through nerve signals through the vagus nerve. As I alluded to, these new obesity treatments are based on these gut hormones, in particular one about which I'll have much more to say, known as GLP-1. Importantly, these drugs did not grow out of efforts to identify anti-obesity drugs. They grew out of efforts to develop new and better treatments for diabetes. And I'm going to tell you about, in the course of this talk, 4 more hormones, either briefly or in greater detail: glucagon, amylin, GIP, and particularly GLP-1. These acronyms don't really matter. They're just different hormones made by different places that have different effects. So these treatments developed out of the need for better anti-diabetic therapies. So here we are with another hormone, one you've all heard about, insulin. A lack of insulin signaling causes diabetes, and similar to what I told you, there are two types. In type 1, you make no insulin whatsoever. As a consequence, it doesn't act on its receptor to lower blood glucose. Blood glucose goes up, and that's type 1 diabetes. Type 2 diabetes, there's no issue in making insulin. In fact, you overproduce insulin because there's a block at the receptor or downstream of the receptor, leading the insulin to not work well enough, which leads to an elevated blood glucose. Now, when insulin was developed, it was thought to be a cure, but over time it became evident that over the long term, insulin is not a perfect therapy for type 1, and it's not at all a perfect therapy for type 2, and so new treatments were needed. And the new treatments that were developed grew out of some work that was done, believe it or not, more than a century ago. In 1906, some scientists, Benjamin Moore et al., about whom very little is spoken, said that, and this is, mind you, before insulin has been even identified, that there's some factor in the intestine that senses nutrient and in turn makes a hormone that lowers blood glucose. And in 1932, based on the work of this man, Labarre, he named that new hormone incretin, which he defined as a gastrointestinal hormone that lowers blood glucose, and by this time he was able to show that it stimulated insulin secretion. And this could represent a new therapy, but with the discovery of insulin in 1923 and the dramatic effect that was observed, an absolutely earth-shaking effect in its day and since, the search for incretin withered. But over time, as it became evident that the available therapies for diabetes were limited and that insulin was an imperfect therapy, interest in identifying incretin was invigorated. I'm going to show you now a slightly more complicated data slide, but it's an important one because it's the single experiment that completely energized the field to find incretin or incretin. Labarre was Belgian, hence the E, the terminal E. Okay, so what's done here is something many of you may have heard about, which is a glucose tolerance test. If they're testing you for diabetes, they give you a glucose load. Usually you drink the glucose, and a normal patient will secrete insulin and their blood glucose will come down very quickly. I'll show you that here in a moment. What McIntyre and Turner did, as well as Dan Kipnis and other groups, was the following. They compared the effect of giving the glucose either directly into the gut orally or giving it intravenously on the increase in blood insulin and the lowering of blood glucose that you see. And what they saw in both cases was that the intrajejunal or the oral glucose was much more effective than the intravenous glucose. That's actually opposite to what you normally expect. Usually if you give something directly into the blood, it works better. That's not the case here. So giving glucose into the gut does a better job of elevating insulin and lowering glucose than it does just injecting it intravenously. This was consistent with the incretin hypothesis, and they concluded that glucose must induce a factor from gut that stimulates insulin secretion. That's what insulinotropic means. And the search for the factor, an incretin, began. And that's why many of you may hear in the current literature that these are incretins. So with this as the starting point, every time a digestive hormone was identified, somebody tested it and asked, does it stimulate insulin? They did that for gastrin, secretin, cholecystokinin, motilin, GIP, and none of them had a durable effect on blood glucose and insulin secretion, at least in diabetic patients. Although I will say more about GIP in a moment. So the incretin lay undiscovered, and it wasn't really even clear that there was an incretin until a set of studies that were initiated based on another hormone, believe it or not, known as glucagon. So glucagon is a hormone that's made in your pancreas, as is insulin, in response to a low blood sugar. In response to a low blood sugar, glucagon is secreted and acts on the liver to produce glucose and elevate the low subnormal levels of blood glucose back to normal. So it's sort of, it counteracts insulin. It's the counterpoint to insulin. Now, all of these hormones are made as large precursors that are chopped up into little pieces. So oftentimes, one hormone precursor will make multiple hormones. And so Joel Habner and colleagues at the Mass General Hospital in 1982 cloned glucagon. What that means is they're able to sequence the gene and decide what the protein looks like and how it's cleaved. And what they found was that in the pancreas, this long precursor is chopped up to produce glucagon, the hormone I just mentioned to you, but in the gut, it's chopped up to make a different molecule called GLP-1. And GLP-1 in this experiment is a 37 amino acid peptide that's the direct cleavage product of this precursor. So, as was the case for all the other hormones that had been identified, GLP-1 was placed on beta cells or given to animals, and the question was asked, does it induce insulin secretion? And the answer was, it does not. The resolution of this mystery, however, wasn't resolved until Svetlana Moisov, working with Habner at the Mass General, an independent investigator there, developed very sophisticated techniques for characterizing the molecules produced by this hormone precursor. And what she was able to show in a very elegant set of studies is that proglucagon, the precursor molecule, is not cleaved to generate the 1 to 37 form. It's cleaved to generate a 7 to 37 form. And now when they put the 7 to 37 form onto cells, insulin stimulation is very, very robust. I'll skip through all of the animal data supporting this and show you what happens in people in a paper published by Nathan, Moisov, and Habner in 1992. In white, you're giving GLP 1 to 37. There's no effect on glucose, and there's no effect on insulin. But if you now give the GLP-1 7 to 37, remarkable reduction in blood glucose, remarkable increase in insulin. GLP-1 7 to 37 is the missing incretin. Now, there was a problem with this experiment, not really a problem, but a fact that limited its commercialization, and that is that the normal molecule is very, very, very unstable. It normally turns over in 3 to 5 minutes. So while you can replicate this effect by infusing it at constant high levels, that's not a viable therapeutic, and there was no way so the quest began to stabilize GLP-1 in plasma, make it longer acting, because 3 to 5 minutes is not long enough to exert a therapeutic effect. So a company, Novo Nordisk, about which you've heard, developed a stabilized form of GLP-1 that they named liraglutide. It was stabilized in 2 ways. They introduced point mutations that blocked its degradation. I'll say more about this in a second. They also added a fatty acid to the molecule chemically. And fatty acids bind to a blood-borne factor, albumin, and they stabilize it. And so with these manipulations, now liraglutide had a half-life of 13 hours. And here's the result of treatment with this. You see a remarkable increase in insulin, lowering of blood glucose, and a lowering in red here of HbA1c, which is a blood marker for how well your diabetes is controlled. So liraglutide was approved, I don't know, probably 15 years ago. Many people have on it. You've seen commercials for it, I'm sure. It's a very effective therapy. But the scientists at Novo Nordisk, in particular Dr. Knudsen, noticed that in addition to improving diabetes, these drugs also reduced weight. Not dramatically so, but certainly significantly. They found that 60% of people who took liraglutide lost more than 5% of their weight, and 30% even lost more than 10% of their weight. There are also some adverse effects, nausea, bloating, other GI symptoms. That might be a clue that they're acting on the satiety system, which induces precisely those responses. In fact, it's now concluded that this reduction in weight that you see is a result of the inductions of nausea, or at least satiety. These are on-target effects that in many cases lead people to either stop the drugs or to tolerate them poorly. But nonetheless, with this result, they decided, well, if 13 hours gives us, half-life gives us this much weight loss, what if we even develop a more stable form? So they made such a form. Now what they did is they took GLP-1 and they combined it not to one, but two fatty acid, dual fatty acid. They made other changes in the GLP-1 sequence. And now they made a drug that had a half-life of 7 days and that's not cleared from the body for 4 weeks once you start it. And now this new drug, semaglutide, which you've heard of on commercials as either Ozempic or Wegovy, reduced weight an average of 16% at 1 year. This is remarkable. A huge weight loss average. But here again, a lot of GI symptoms. 44% of patients had nausea, vomiting, or constipation. In some cases, these drugs can also induce gastroparesis. They sort of lead to a destimulation of your stomach. And actually, some patients now are told by surgeons, you need to stop these drugs before you have surgery because the stomach contraction is inhibited. There are some anecdotal effects. They've not been rigorously assessed. I don't think there's any reason right now to worry about things like suicides that have been reported, but experience with the sequelae of these drugs will undoubtedly grow. Okay, so what I want to emphasize here, though, is that this effect is what we refer to as pharmacologic. This is not a physiologic effect of the drugs. GLP-1 normally doesn't do this, but if you give it at a high enough dose, it suppresses food intake and weight. And part of the reason we know that is that mutations that reduce GLP-1 signaling don't affect weight at all. So these are not physiologic regulators of weight. They don't cause obesity or leanness, but high enough doses of it co-opt other systems to reduce food intake by activating the satiety system. We, of course, need to know where the hormone acts to perform its various tasks. And we now know that semaglutide acts normally to regulate insulin secretion in the pancreas. But when you give it at high enough levels, as was the case for these stabilized drugs, it acts directly on brain circuits that regulate satiety and appetite. Well, the story doesn't stop there. A chemist and biologist, Richard DeMarchi and Matthias Chup, started to now ask, can we make a better form of GLP-1? And here come in some of the other hormones I mentioned. These are the amino acid sequence of glucagon. This is GLP-1. This is GIP. And what they started to do is mix and match, introduce changes so that now a single molecule will not activate only its one receptor, but it might activate 2 or even 3. So they made single molecules that could both activate GLP-1 and glucagon signaling, or GLP and GIP signaling, or in some cases GLP, GIP, and glucagon signaling. And now these molecules have been tested in animals and then in humans, one of which you may have heard about is called tirzepatide. This is a GLP-1 GIP dual agonist made by Eli Lilly. It was built on the work of DeMarchi and Chup. And what you can see here is each amino acid in the peptide chain is shown in a different color, and it has some GLP peptides, it has some GIP peptides, it has some new peptides, and so on. And now this dual agonist is referred to, because it targets 2 receptors, has an even greater effect to induce weight loss. 22% versus 15%, with even less nausea. They seem to be better tolerated for mechanisms that are being uncovered by a scientist named Steve Lieberlees and others. So where are we and where do we go from here? Well, these new treatments have unlocked the door. There are intrinsic difficulties in developing new drugs, but once you have a first effective drug, it opens the door to new drugs. And this may include in time one of these triple agonists that I showed you, the GLP, GIP, glucagon triple agonist known as retatrutide, which is in clinical development and gives even more significant weight loss. But these drugs are not a panacea. There are still unmet needs despite the breakthrough status of what are referred to as these incretins. First of all, as I mentioned, there are tolerability issues due to nausea, other GI side effects, as well as gastroparesis and perhaps other problems. In addition, as weight is lost, you lose lean mass. That's true of any type of weight loss. Lean mass is muscle loss. But importantly, even if you stop the drugs, weight goes back up. You need to take the drugs as a lifelong therapy, but the loss of muscle mass doesn't fully recover, or doesn't appear to fully recover. In addition, not everybody responds, either for reasons of tolerability or other reasons. And that's shown here in the tirzepatide trial. I'm not going to go through this at any length, other than to say these are the patients that not everybody responded. That's shown here. You're not at 100%. And about 30 to 50% of patients don't receive what their target weight loss is. In addition, as I mentioned, you have to treat yourself with these drugs as a lifelong endeavor. And so there might be a need in time for something that doesn't induce the weight loss but might maintain the weight loss. So you can go off these drugs but take something else to stabilize the new lower weight. And I predict that in time there'll be, similar to other disorders, there are going to be tailored therapies for different people with different pathologies. So I want to close with two slides. First, by analogizing for you, I think, obesity in the future to diabetes. Diabetes comes in two forms. Type 1, you make too little insulin. Insulin therapy is a pretty good therapy here. Type 2, you make lots of insulin, it just doesn't work well enough. For this, alternative therapies are needed, and that's where liraglutide came in. Similarly, for obesity, I think that some patients make either no leptin, as I showed you, or too little leptin as a result of just defects in synthesizing the molecule. Well, leptin may be a pretty good therapy for that subset, the size of which is not known. But most patients have leptin resistance, and here the incretins seem to work. Incretins plus leptin may have a synergistic effect in people, we'll have to see. But there may also be ways to modulate the neural circuits on which these hormones act. And in the last slide, second to last slide, I'm just going to tell you that an enormous amount of work, basic science work of the sort that I think will be in part performed at the new institute here at Cold Spring Harbor, is mapping the neural targets of all these different hormones. The satiety hormones act on a part of your brain known as the brainstem. We know something about where they act, but not everything we need to know. We've heard a lot of talks at this meeting about signals that regulate the reward pathways, and I told you about leptin modulating the homeostatic system. And there are new methods now that allow you to pick out cell-specific drug targets, some of which are in development at Cold Spring Harbor here, that modulate these activities of the neurons. And in the future may even include small molecule drugs that target one cell type or another as part of this ever-expanding neural circuit that regulates weight. Well, of course, as always, questions remain. We know that, or we believe incretins act primarily on the brainstem. We don't know the precise cellular targets. We need to. We don't know why leptin synergizes with these gut peptides. In time, we'll learn what the long-term effects of these drugs are. They are circulating at enormously high levels in patients. I think we'll need means to try to prevent the loss of lean mass, or at least allow it to recover. And then I think the future may also hold the promise of drugs that modulate specific pathways. So I'll close here and try to summarize what I tried to communicate today. Defects in the leptin-melanocortin system cause obesity. 3 systems control food intake: a homeostatic system, a reward system, and a satiety system. New therapies for obesity were developed based on drugs for diabetes, and these have pharmacologic effects to induce satiety, and that new therapies, I believe, are forthcoming, some of which will be tailored for different individuals, others of which I think in time may help us treat the root causes of obesity. So I'll stop here. Thank you all so much for this opportunity and your attention.